Welcome to this video on lateral flow immunoassay. In this video, we'll explore what lateral flow immunoassay is, how it works, and its applications in detecting different diseases. We'll also discuss some of the advantages of lateral flow immunoassay and the current state of the field. Let's get started. Lateral flow immunoassay, also called LFIA, is a technology behind low-cost, simple, rapid, and portable detection devices. Actually, most of the COVID-19 antigen rapid tests use this technology. So during the pandemic, the vast majority of people have used flow immunological analysis products. COVID-19 antigen rapid test strips have a control line and a test line. The control line should appear as a colored line regardless of the test result. If the control line does not develop within 20 minutes, the test result is considered invalid and retesting should be performed again. When the test line appears as a colored line, it means that it has detected the COVID-19 antigen. The subcomponents of a lateral flow immunoassay use specific and optimized components. It can be in a dipstick or cassette format. All lateral flow tests consist of 3 main pads plus a membrane. These different parts in order are the sample pad, the conjugate pad, the membrane, and the absorbent pad. Lateral flow immunoassay can be configured to work with a range of liquid samples, either whole or prepared, including saliva, blood, urine, serum, milk, plant material food, and so on. Let us take a look at what happens when we apply a sample to a lateral flow immunoassay. First, the sample gets added to the sample pad, promotes, and distributes the sample evenly towards the conjugate pad. In the case of lateral flow immunoassays, conjugate pads usually have labeled monoclonal antibody, which has been dried directly onto the pad. Once the sample reaches this pad, if the target analyte, i.e. the sought-after substance, is present, it binds to the monoclonal antibody. The labels give color to the control and test lines. Labels are usually colloidal gold, carbon, or latex. Labels are chosen on some compatibility with test components. Then, the sample molecules, some of which have bound to the antibody, flows to that membrane, which is usually made of nitrocellulose. It is essential that the membrane possesses good capillary forces and ease of binding. Here is where both the test line and control line is located. The test line is the first of these lines and is coated with monoclonal antibodies that are also specific to the antigen of the target analyte. This means that if we can detect a signal at the side of the test line, the target analyte has successfully bonded twice to 2 different monoclonal antibodies specific to it. Then, the unbound labeled antibodies continue to the control line where they bind to a secondary antibody. Thus, a line from their label forms here as well, and this allows us to check that the test is not faulty. Finally, the sample flows through to the absorbent pad. The primary function of the absorbent pad is to wick the fluid through the membrane. Cellulose filters are commonly used as the material for these absorbent pads. When the absorbent pad is present, the amount of sample can be increased, improving the sensitivity of the test. This is because it absorbs excess sample. Totally speaking, the principle of LFIA is based on the use of antibodies or antigens that are immobilized on a nitrocellulose membrane. The sample is then added to the membrane and allowed to flow through it by capillary action. If the target analyte is present in the sample, it will bind to the immobilized antibody or antigen and form a complex that can then be detected by a labeled antibody or antigen. The labeled antibody or antigen will bind to the complex and produce a visible signal that can be read by eye or with a reader. Lateral flow immunoassay tests typically complete within 2 to 15 minutes. Tests may be read visually or with a LFIA reader, so it is fast and easy to use. With this technology, companies can come up with more affordable assays for different use. The adequate analytical sensitivity, together with versatility and good usability, make lateral flow immunoassay the most commercially available POC diagnostic format. This is also due to the fact that, in contrast to other promising paper-based analytical platforms that mainly remain as laboratory prototypes, lateral flow immunoassay devices easily enter into real-life applications, with a high market penetration mainly because they do not need extensive upgrades to become an end-user device. Nowadays, lateral flow immunoassay is a widely used point-of-care method for both diagnostic and environmental applications. LFIA can be considered as a sort of lab in a hand, and together with other point-of-need tests has represented a paradigm shift from sample to lab to sample, aiming to improve decision-making. Before the pandemic, when we talk about lateral flow immunoassay tests, people come out with pregnancy tests. After the pandemic, when we talk about lateral flow immunoassay tests, people come out with COVID-19 tests. But that same platform where you see one or two lines to indicate a yes or no can be applied to a lot of different things. For example, it can detect infectious diseases like malaria or HIV, and establish whether a patient has an allergy by detecting total IgE or allergen-specific IgE. For example, the HIV-1/2-AB plus combo rapid test from CTK Biotech is a lateral flow immunoassay for the simultaneous detection and differentiation of HIV-1 and HIV-2 antibodies in a human serum, plasma, or whole blood. The product only requires 20 microliters of specimen. Results can be available within 15 minutes to allow prompt action to be taken at the point of care. Lateral flow immunoassays are also being developed for numerous disease biomarkers, including prostate-specific antigen, which is often elevated by prostate cancer and other prostate disorders, and C-reactive protein, a major inflammatory marker. Outside of patient care, other applications of lateral flow immunoassay include screening for animal diseases, monitoring water quality, and safeguarding food production by testing harmful contaminants such as bacterial toxins. After about half a century from its first development, the lateral flow immunoassay technique is increasingly used thanks to its global scalability and to its features that allow reducing costs and workload, while enhancing the workflow efficiency, improving the turnaround time. Lateral flow immunoassays have become one of the reference point-of-need tests to obtain results in a timely manner without the need of high sophisticated and high-cost laboratory equipment. The interest of the scientific community for the lateral flow immunoassay is still very high, as it can be noted by the increasing number of research papers aiming to improve one or more aspects of the technique. The sensitivity enhancement and the quantitative output have been the most studied features during the last decades. Nevertheless, the colorimetric detection is still the most widely used detection method, and gold nanoparticles can dominate the colorimetric label scenario. In conclusion, lateral flow immunoassays are powerful technologies for diagnostics. In the future, we may find more improvement in this technology, maybe the new detection labels, the new functionalization methods, or the new conjugation techniques.